So thank you, everybody. Thank you, team. It's always a pleasure to talk to you guys. And as you can see from my title, I'm going to take this opportunity not only to talk about what the lab has been up to in the last 6 years, in case you're wondering, but also I want to make an appeal for the use of multi-level of hierarchical models in ecology, because we have to deal very often with what we call complex data, but what it is just messy field data. And this type of modeling approach is very handy to deal with many of those issues. Before I start, I want to set up the expectations for this talk very clear, so if you don't like it, you can leave now. It has to do a little bit with the process of science, too. So when we start our research projects, we usually have a question that we think is relevant or interested. And then we design a way to collect observational data, or we design an experiment that we carry out. And after a few years of very arduous work, if we are lucky, we may be able to quasi-answer that question. And I don't know about you, but I've never been able to fully answer the questions I started with. But what is going to be always true in any of these experiments is, by the end, we are going to end up with many more questions we started with. And this somehow represents also the process of science. It's always about discovering what else we don't know. So with that, what I'm going to be doing today is to start with a defined question that I'm going to be trying to answer. I won't be able to answer it, but I probably will leave you with many other questions that are still unanswered. So let's start. Forest ecosystems. These are completely fascinating ecosystems, a pleasure to work in, and also a very good example of a good story of how we can use our ecological knowledge to conserve them, also to be able to restore them, to enjoy them, and even if you don't believe me, in some cases we can also extract resources in a sustainable way. So if you think about successional dynamics, disturbance dynamics, species interactions, we have been using this knowledge that we have from forest ecosystems to both conserve and manage them. And this has been working pretty well in the last century, but now in the last few decades, what we have seen is that the rules of the game have changed. Now we have global change. And this is not just about climate change, but it's also about changes in landscape configuration, changes in the species that are around, many new introduced species. Also pollution are affected these ecosystems. So what we have now is this synergistic effect of many drivers of change that may be affecting that dynamics. So we have a disturbance in one of these forests. What kind of successional pattern should we expect? Is what we have studied in the past, or is it going to be something new? We don't know, and we haven't been able to answer that question yet. And if there is an area of research where most people agreed we are lacking any information, it has to do with how biotic interactions may be affecting the response of organisms to global change factors. And I just brought here an example of 4 papers that have been published in the last, I think, 3 years, pointing at that lack of knowledge. So if we think about short-term dynamics to those global change drivers, biotic interactions may play a very important role, and we know very little about them. So this has been the topic of the lab. We have been studying how different types of species interactions, biotic interactions, may be affected forest responses to global change. And today I'm going to be focused on one of those particular interactions, and that's plant-soil feedbacks. So a few years ago, Sarah McCarthy-Newman and we started a series of experiments where our objective was to try to quantify how these plant-soil feedbacks may be driving the colonization dynamics of 3 species tracking global warming. And before I start, I just want to explain really briefly what are plant-soil feedbacks. So plant-soil feedbacks are going to take place when a plant growing on one particular site, through the exudates of the roots and decomposition of the litter, is going to be changing the physical, biological, and chemistry characteristics of the soil. And those changes are going to affect back the plant community. So if you think about that from the point of view of a seedling that is trying to establish under the canopy of adult trees, that particular seedling is going to be affected by all these feedbacks. And these feedbacks can be biotic, but they can also be abiotic. They can be beneficial, like mycorrhizal fungi, and I'll be talking a little bit about that. But they can also be negative, like pathogens, and I will also talk a little bit about that. So again, it will be the combination of all those feedbacks that will determine if that particular tree species can establish or not under the canopy of those adults. So if you think about how plant soil feedbacks may play a role in tree range expansion, what we did is to develop two kind of opposite hypotheses. And these hypotheses are the extremes of a gradient. So don't think like it has to be one or another. It could be anything in between two. So for hypothesis one, what we thought it could happen is that when these species are establishing beyond their range, maybe because they are naturally migrating in response to global warming or because we are moving them to areas that are warming, what it may happen is that they may be released from their host-specific pathogens, the pathogens that are associated to the soils within their range. At the same time, if they are able to benefit from the mycorrhizal fungal symbiosis that they need to grow, what we could expect, at least in the short term, is a greater dominance of these migrating species. Because as it happens with invasive species, they will be released of their natural enemies, while at the same time may still benefit from the symbionts they need. The second hypothesis, which is the other extreme of that gradient, will be the opposite scenario. These cilians are trying to establish in a new range, and they may still be affected by soil pathogens. So there is no enemy release. They still have to deal with those pathogens. At the same time, they may encounter suboptimal conditions, and here I'm going to define them as they may not be able to find the right mycorrhizae or the most optimal mycorrhizae for that symbiosis, which means they may not be as competitive as they could, and they still have to compete with the local community that will have the right mycorrhizal symbiont. So what it may happen here is that those cilians of those colonizing species may not be able to establish in numbers that are large enough to develop a viable population, so colonization won't be taking place. So with these two hypotheses in mind, we start designing our experiments. And we were working here in the Great Lakes regions, and this is one of my favorite slides, although I'm not sure if you're going to be able to see what I would like to show you. But if you look here in the middle of Michigan Lower Peninsula, there's a change in colors, darker in the north, lighter in the south. That change also takes place here in Ontario and also here in Wisconsin. And what that line is reflecting is the northern distributional limit of commercial agriculture. But that's also the northern distributional limit of many tree species. So we have a perfect experimental design here in the Great Lakes because we can work within the range of the species and also beyond the range of the species. So to answer all the questions we have, we designed a series of experiments both in the greenhouse and in the field. And even if we didn't intend it that way, it was a good choice of having both of them because both types of experiments have cons and pros. So they were very complementary with each other. So greenhouse experiments are very good if you want to focus on the mechanism or on the interaction between two particular variables. You can control everything and only modify one factor. So these experiments were very useful for us to be able to develop the analytical models that we were going to use to analyze demographic data for these plants. The problem with greenhouse experiments is that the demographic rates that we got, the survival and growth of these cilians, were very unrealistic. They don't have anything to do with what we find in the field. So we can't really use them to make any kind of comparison among species, neither to make any kind of predictions of what we could expect under different conditions. But for that, we have the field experiments. So the field experiments allow us to plant the same species in the areas for which we want to make predictions. So we were able to obtain demographic rates that are very realistic of what is happening in the field, demographic rates that we could compare among different species and that we could also use to make predictions. The problem with field experiments, if you have worked in the field, is just you can't control anything. It's practically impossible. It doesn't matter how much you try it. It doesn't happen. But for that, we have the greenhouse experiment. So the way we went about it is by dividing our region in what we call south area or within the range, and then the north area and beyond the range. And for both the greenhouse experiment by collecting soils and the field experiment by working in field sites, we studied different species of seedlings, different species of tree. And for all the seedlings that we planted, we collected similar data in both the greenhouse and the field. So we collected data about the abiotic environment they were growing under. So we have soil moisture, light, and soil nutrients. We also collected data on the seedlings, survival and growth rates, and also mycorrhizal colonization of the roots. So this is a little sketch of what we did with the greenhouse experiment. Here, what we did is to collect soils that have been cultivated under the canopy of adult trees. So we selected 8 different sources. These were 8 species. 4 of these species grew both in the north and the south, and we collected soils from both the northern area and the southern area. And then we chose also another 4 species that only grow in the south, and we collected soils from under the canopy of adults. Half of the soils were just kept at control. The other half were sterilized with gamma radiation. And then we planted germinating seeds of all these species in all combinations of soils and grew them under both control and sterile soils. And then we grew the seedlings under 3 different levels of light. And these are realistic levels of light that you find in the field. And the reason why we added this component is because light is a major limiting resource during the recruitment of all these temperate tree species. So species have evolved to deal with different levels of light and respond in different ways. So including light was an important component in our experiment. So we grew these seedlings for 10 weeks. We collected data in their survival during those 10 weeks. And then for those seedlings that were still alive, we collected data on growth rates and mycorrhizal colonization. So this is an example of the data for one of the species, Acer rubrum. And here's again, percent survival. We start 100%, how it changes over time during the 10 weeks. Different lights are different soil and sterilization combinations. I purposely didn't bring the legend because I don't want you to start trying to figure out what is here. What I want to show here is how dynamic these survival curves are. They cross each other at different paths. So if we were to use a classical survival analysis to analyze this data, it will give us what is called median survival time. That is something what is happening somehow around there. But it will miss all the dynamics that we see here. It will also miss a lot of the individual information that we have for each seedling. So instead, what we did is to have an individual-based model where we will analyze every individual seedling. And we will do it as a function of what we are interested, the soil source, the region, the treatment. But we also have information on the seed side at the time of planting. And that's very important because maternal effects may be driving survival of the seedlings. And we also have information on the particular soil moisture and light that each seedling was receiving. So again, we'll be able to customize our analysis for each of the seedlings. And these are some of the results, again, for Acer rubrum. So these are results for seedlings that were grown at low light. And this is important because the dynamics that we saw, it was different if it was at low light or high light levels. So here, what we have is changes in survival. So given the average survival for this species, how much it changes with respect to the soil source, which is what we have here. And we have that for non-sterile soils in black and sterile soils in gray. So as you can see here, there are quite big differences in survival depending on the soil. So there are soils in which Acer rubrum survival was very high, like right here. But there were soils where survival was very, very low. And there is a whole ranking of soils. And this is an important factor because especially after last week's seminar with Steve Hubble, he was a little bit talking about conspecific soil versus or conspecific pathogens versus heterospecific pathogens. And that's very common in the plant soil feedbacks literature that you just compare between conspecific and heterospecific. And my point here is that it's not just heterospecific. There is a whole range of variability in survival rates depending on the soil. So this adds a new dimension on the recruitment niche of these species. It matters where those seeds are falling. Not all soils are equal. So we have this result for all the species. How the soils rank, it varies one species to another. They are not general patterns. The only general result that we got is that for 7 out of the 8 species, the worst soil was always the conspecific soil. In this case, acer rubrum collected in the south. So it is true that there are strong conspecific effects, negative effects, but again, other soils also matter. To test if these effects of the soils were biotic or abiotic, then we make those comparisons with a sterile soil. And here, at least for 3 soils, the ones with an asterisk, we saw statistical significant differences between growing in a sterile soil or controlled soils. For 2 of them, the conspecific soil and Quercus velutina soil, sterilization increased survival. So what it means is that here we have a negative biotic effect of the microbial community in those soils. In the case of Robinia pseudoacacia soil, this one here, we saw the opposite effect. Sterilization decreased survival. So what we see there is a positive biotic effect of those soils. So what happened with those northern soils? So at least for this species, as you can see here, they just rank in the middle. So this is a species that grows both in the north and the south. These seeds were from the south, but as you can see, northern soils were not particularly better or worse. This was common for all the species, even the ones that only grow in the south. There were no differences, at least no pattern that northern soils were different from southern soils. We also collected data on growth, and this is data for another species, Acer saccharum, sugar maple. This is the data for all the non-sterile soils. And as you can see here, there is a response of growth rates to light. So all these species respond quite well to light. So under higher light levels, we see higher growth rates. And even if you don't believe me, this is a very clean data on growth. Usually you just have a non-descriptive cloud of points. It doesn't tell you anything. But here, because it was a greenhouse experiment and we control for everything else, we see this clear response to light. But what we also see is a lot of variability in that response. So the challenge here is, can we explain all that variability? And here is where I'm going to be doing my first appeal for hierarchical or multi-level models. So when we model growth, we always, or almost always, use this asymptotic curve where we estimate growth as a function of a limited resource, which in our case is light. And we use this asymptotic response that is described by 3 different parameters. So growth is equal to Gmax, which will be the maximum growth rate that the species can reach when resources are not limited. Then we have a second parameter that I call here L0. L0 is the amount of light necessary to start growth. So here is where photosynthetic rates are higher than growth rates and the plants can start growing. So this is a very important parameter because it tells us a lot about the competitive ability of these species under low light conditions. And the third parameter is this curvature parameter here, theta, which it tells us how responsive a particular species will be to an increase in light. Can it respond fast and grow to its maximum potential fast, or is it going to be a low response to higher light levels? So again, first attempt to convince you that hierarchical models are very useful. So first of all, definition, what is a hierarchical model? And I'm just going to keep it really simple. So you can call a hierarchical model a model where your parameters are going to be treated as random variables, and they are going to have their own parameters and their own models. So in our particular case, what we have is our growth data. Right here, we have defined the process, which is that equation that I just described that has 3 different parameters. And now what we are going to do is to give those parameters their own models that will have their own parameters, which usually we call hyperparameters. So what this allows us is to introduce a lot of information that we have about these sealants that can explain all that variability that we saw in the data. And this is how we went about it with respect to the modeling of the parameters. We modeled these 3 parameters as a function of what we were interested in: soil source, the region, the treatment, control or sterile, also the seed site to control for maternal effects, soil moisture, soil nutrients, all the things that we know are going to be affecting growth. And also we have information on mycorrhizal fungi colonizing each of those sealants. And the reason why I introduced this factor, colonization by mycorrhizal fungi, is because when we think about the mycorrhizal symbiosis, we always think about being positive, plants always benefiting from it. But the truth is that this is a relationship that depends a lot on the surplus of carbon that that particular plant may have or not. So when there is plenty of resources, in this case light, the plant is fixing enough carbon that it can both grow and feed the mycorrhizal fungi. But if this symbiosis is taking place under very low light conditions, the plant may not have enough carbon to both grow and feed the mycorrhizae. And what we see here is more of a parasitic relationship and not a mutualistic one. So we analyze the data that we have in growth, including all those parameters I talked about. And these are the results for Acer rubrum, Acer saccharum, the species that I just showed the data for. This is the specific results when growing in its soil, own soil, the conspecific soil. And here again is the growth rate, predicted growth rate as a function of light. And we have 2 types of seedlings: the ones that develop the mycorrhizal symbiosis and the ones that didn't. And these all were non-sterile soils. So what we see here is that in solid lines, this is the response to light of those seedlings that develop the mycorrhizal symbiosis. This is the mean and the 95% predicted interval. And this is the response of the seedlings that did not develop the symbiosis in those 10 weeks. So what you can see here is that there is a dynamic that changes along the light gradient. Under very low light conditions, seedlings that are grown without mycorrhizae have higher growth rates than those seedlings that have the symbiont. So what we see here is a parasitic relationship. Then it crosses a light gradient, after which the 2 intervals overlap. So at least statistically, we cannot say they are different. And I call this area an area of commensalism. It's more of a neutral relationship. The plant doesn't gain or lose for having the mycorrhizal symbiont. And then again, it crosses another light threshold right there. And here it's very clear how plants that are growing with mycorrhizal fungi are growing much more than plants without it. So here we have a clear mutualistic relationship of those seedlings. So we run this analysis for all the species in all the soils, and we see different dynamics. This is one of the most beautiful ones, as you can imagine, because you can see the 3 types of associations. But for some of them, they start as parasitic and they only move to commensalism. Other ones, maybe they start here and then just move to mutualisms. Others may be neutral for the whole light gradient. We didn't find any particular patterns with different soils and different regions. So we couldn't address any of the questions that we have about it. But seeing this dynamic generating many more questions. Well, if there is this kind of dynamics where the relationship changes, what it will mean if this particular species were to grow beyond each range and it couldn't find the right mycorrhizae, the mycorrhizae that is more affine to developing the symbiosis. So here I'm speculating completely, but imagine that what these 2 scenarios represent now, these will be the seedlings grown within the range and the seedlings growing beyond the range of the species. So if this is what it may happen, when this particular species will try to colonize a new area, what we will see is that under low light conditions, if they don't develop the mycorrhizal association, we'll see something to what we speculated in our hypothesis 1, enemy release. because they will be able to grow more than they would within the range with the mycorrhizae. But if those seedlings were to be growing under high light conditions, we'll see the opposite pattern. They will be growing at much lower growth rates than those within the range with the right mycorrhizae. And we will be seeing here is suboptimal conditions of these seedlings. So as I said, this is just a speculation, but these are some of the new questions that we have after we carry out those experiments. So just to wrap up this experiment, the greenhouse experiment, we didn't see major differences in survival and growth between northern and southern soils in any of the species we tested, so there were no major differences. The only general pattern that was very clear, it was the negative effect of those context-specific plant soil feedbacks, and that only took place under low light conditions. So the environmental conditions really play an important role determining the extent of these biotic interactions. How each soil affected each species, that ranking that I showed you a few slides ago, it depends on each species. So each species has its own niche of recruitment dynamics of which soils are the best one or which ones are the worst one. And then when we study the nature of the plant mycorrhizal fungi symbiosis, we saw that it changed along the light gradients, and here is where I speculated that it could play a role on range expansion depending on both the species being able to generate that symbiosis or not, and the environmental conditions it will be doing it, high light or low light. So this experiment was very illuminating, and we really enjoyed it because it told us a lot about mechanisms. It allowed us to work a lot on those models for growth and survival. But as I said at the beginning, those survival and growth rates that we measured were very unrealistic. So we couldn't really use that to make any inference of what it may happen in the field. So for that is why we have the field experiment. Here we were able to work with many more species, and we divided them into groups. The group that we call the northern group, or local species. These are species that all grow in the northern area, and they also grow in the south, and for some of them we use seeds from both the north and the south. Then we have a second group. These are our southern species, or the migrant species. These are species that only grow in the southern part of the region, but these are species that are predicted to move north as climate warms. This is a little sketch of how the experimental was carried out. So we planted seedlings that were a few weeks old of all those species in 2 regions, the north and the south. Within each region, we look for a gradient of soil moisture availability for the plants. So we have different stands, and the intention was to go from very wet to very dry. Within each of those stands, we planted seedlings into habitats under the forest canopy, so low light conditions, and also in open spaces. And then there was one more arrow here, which was a fungicide treatment, because we wanted to look at these plant soil feedbacks, but it completely failed. So I decided to erase it because, as I said before, you can't control anything in the field. So it didn't work. So we planted these seedlings and we followed them during the first growing season. We collected data on both survival, growth, and colonization, and also colonization of the roots by mycorrhizal fungi. So this is the experimental setup, but also this is the structure of the data. So the data that we have was collected for individual seedlings that were in different habitats. Those habitats were nested in different stands that were set up in different regions, and then we have different species. So this is the structure of the data, but here again is where hierarchical models can be very handy, because we can represent the structure of the data also in our analysis, and we can estimate parameters at all these levels. We can estimate parameters at the individual level, habitat level, stand, region, and species level. We can do any kind of combinations. We can skip any level. So what a hierarchical model gives us is a lot of flexibility in the analysis of our data that allow us to make many more comparisons that we could with other methods. So this is just a summary of the survival data for all those species, how survival changes from one region to another. This is the predicted survival. We use those models that we develop in the greenhouse experiment and apply them with our data here. And here, this is the group of northern or local species. This is the group of the migrant species. And this is predicted survival, the mean and the 95% predicted interval for the north and the south. So you see all of them together. There are differences species to species, but you cannot say one group is better than the other. So again, pretty similar. The only thing that is a little bit clear is that it looks like survival was slightly higher in the north. And for a few species, the one in blue, that was statistically significant. So these species in both groups grew better in the northern sides. And we have a couple of species, the ones right here in red, where survival was higher in the south. We also have growth data. This was extremely messy data, very difficult to analyze. But again, we had the models that we developed in the greenhouse. And I just brought here one slide to show you how we couldn't find anything on this analysis. So this is just the results for Gmax, the maximum growth rate from that type of asymptotic curve analysis that we did. So here's again the mean and the 95% predicted interval for all the species, north in black and south in open circles. And as you can see, everything overlaps, so we couldn't figure out anything that was going on here. No differences among species, which is very strange. No differences between regions. But still, we have a lot of data on those ceilings that it was still very relevant. So I talked a little bit about in the beginning how we want to use this field data to make predictions. And one of the most important predictions we want to make is how these species perform along environmental gradients, because that's what is going to allow us to predict responses to climate change. So here, I just brought you some of the data. This is data for Keria glabra, picnic hickory. This is percent of ceilings that were still alive at the end of the first summer, and they are plotted as a function of light. And here in black are plots that were in the canopy, and in gray plots in the gap. And as you can see here, a lot of variability. So there are plots that have pretty much with the same light level, no survival at all, and then plots that have almost 100% survival with the same amount of light. Also important here is that the gap plots are as dark as the canopy plots. So it's important that we measure light, that we are not just using this dichotomous variable or categorical variable, because conditions are very specific of each plot. So here, we see a lot of variability here, and does it mean that this plant, this particular species, doesn't respond to light? I mean, it would be possible. It's a large seeded species, so maybe it doesn't need any light. But we know from the greenhouse it responds to light. So the challenge, again, is trying to explain all this variability that we see in this response. So if we were to analyze it just following a logistic regression model where we are estimating survival as a function of the environmental variables, and in our case we have light and soil moisture, these are the results from that model. This is the mean prediction and the 95% credible interval around it. And the most problematic issue here is not like there is a negative response to light. I mean, that could be possible. Probably not because the plant is physiologically responding in a negative way to light, but maybe it's probably because these high light ecosystems are also very competitive ecosystems. There are a lot of grasses right there, so this is probably what we are seeing here. But what I find problematic here is how narrow this interval is. It's not reflecting the variability in the data. So again, we still have no clue what may be going on, even if we can even analyze it that way. And this is something that I know John was complaining about, that of how models were becoming, they are statistically significant. So these parameters of these models are probably statistically significant, but the R-square is very low. And that's something that bothers me quite a bit. So how can we get the R-square of this model to be much higher, which means we have much higher predictive power? And here again, we have a lot of information of these ceilings, so let's put it all together. And this is my third appeal for hierarchical Bayesian models. It gives us a framework to put together a lot of different sources of information. So what we did in this particular case is, again, to estimate survival as a function of the environmental gradients, soil moisture. Then we divided light in the canopy and light in the gap, because it may respond differently if light increases in the canopy versus in the gap. But then we have also information on other variables associated to these seedlings. We also have growth rates, and we usually associate higher growth rates with higher survival, although we wanted to be sure that was the case, so that's the reason I have a plus sign here. We also estimated growth rates as a function of the resources, so moisture, light, and nitrogen. Growth rate was also estimated as a function of initial plant size to account for maternal effects. As we saw in the greenhouse, mycorrhizal colonization was also important, so we included it there. And then we estimated also survival as a direct function of mycorrhizae abundance, which again could be positive or negative. And then we also have a submodel that estimates abundance of mycorrhizae as a function of the environmental conditions, light and soil moisture. So we ran these models. These are the results, and I'm aware this is the most horrible graph that you have seen probably in a long time, but give me a minute just to show you what I think is an interesting aspects of this graph. So there are pluses and there are minuses indicating positive and negative associations. There are statistically significant in black and not significant results in gray. This particular species was colonized by the two types of mycorrhizal fungi, arbuscular mycorrhizal fungi and ectomycorrhizal fungi. And yes, as I said before, what is, I think, interesting from this graph? So if we look at the direct effects of the environmental conditions, so soil moisture and light, I mean direct in the sense of there are direct effects in the model. What we see here is all negative effects for all of them, as we saw in that null model that I showed you before. So we have here a negative effect of increasing water and light. Then what we have here is the effect of growth. So higher growth rates were associated with higher survival. But at the same time, higher soil moisture and light was associated with higher soil growth rates. So what we have is, through growth, an indirect positive effect of increasing water and light. Next step, ectomycorrhizal fungi. So for that particular species, ectomycorrhizal fungi have a positive association with survival. But when we model the effects of the environment in the abundance of that mycorrhizae, we saw that they were all negative. So through ectomycorrhizal fungi, what we see is a negative effect, indirect effect of increasing water and light. And finally, the association with arbuscular mycorrhizal fungi. This was negative, so more mycorrhizae, lower survival. At the same time, as we saw with ectomycorrhizal fungi, more soil moisture and more light, less abundance of these particular fungi. So here what we see is an indirect positive effect of increasing water and light. So it gets really complicated, direct, indirect effects, positive effects, negative effects, but all playing a role on survival. So how, what is the result of all this? So these are the predictions for that model, the predicted survival. So here again is the predicted probability of survival as a function of light. The dots are still the data. This is still our null model, this one right here. And these are the predictions in gray for the gap and in black for the canopy. And these lines in the middle are the mean predicted growth rate. And these ones at the top and the ones at the bottom reflect the 95% predicted interval. So a very large variability in our predictions, but a variability that is explaining most of the variability in the data, and now we can explain where that variability is coming from. So it gives us a lot more predictive power. I mean, we still have room for improvement. There are quite a few points there that we are not able to explain yet, but I hope you will give it to me that our model is a little bit better than this one here in predicting what is going on in the field. So to wrap up this part, what we saw in the field with our very messy data is that survival and growth rates were very similar among the local and the migrant species. We saw slightly higher survival in the north. The growth rates were similar, but no differences between the two groups. And then when we were modeling the recruitment dynamics along those environmental gradients, we saw how biotic mediated interactions could be important to describe that nonlinear recruitment dynamic that we saw along the light gradient. So now to wrap up and come back to my original question, do plant sulfate bags, will they play a role on colonization of tree species in response to global warming? So as I promised you at the beginning of the talk, I can't really answer that question, even if we try. We have a little bit of an idea what it could happen. So with respect to pathogens, what we see here is that it looks like a large proportion of species may be released from conspecific soil pathogens. So just because these species will be colonizing areas where adults are not present, they may experience a temporal enemy release that will allow them to germinate and establish in higher rates. We also look at what was the role of mycorrhizal fungi, and we were able to quantify that role along environmental gradients. And as I speculated before, you put it in the context of colonization, what we could see is that enemy release under low light conditions, but if those species were to grow under high light conditions, they will be not growing to the maximum capacity, so they will have this suboptimal performance that may not allow them to be competitive enough to establish in one region. And then we have all the field experiments that allow us to make comparisons between those two groups of species, the migrant species and the local community in the northern sides. And what we saw here is that migrant species could be competitive enough, at least during the establishment process, during the first growing season, to be able to establish along the local communities in the north. And with this, thank you, everybody, and I'll be happy to take your questions. I didn't leave you with any questions. Yes.
So in your greenhouse experiment, you were observing colonization by mycorrhizae. That wasn't a treatment. That wasn't a treatment you were applying.
It wasn't a treatment. No, it was. How did we observe the mycorrhizal colonization of the seedlings in the greenhouse experiment? No, we did not inoculate seedlings with mycorrhizae. We let them to be colonized by the natural communities that were in those soils.
So was there any effect of your treatments on that colonization in the greenhouse experiment?
You mean the treatment, the controlled treatment?
Yeah.
The sterilization treatment, there was no colonization at all in the sterilized soils.
Oh, about light?
There were light effects.
Did the amount of light that you were growing the plants under affect the colonization?
It did. Yeah, it did. So that was part of, I didn't show the whole model, but part of the model is also, it has two parts of it. The first one is colonization probability, and the second one is the abundance of mycorrhizae, and both of them are dependent on the environment. And colonization started at different light levels in different species and different soils. Yes?
In the field experiment, did you have different species growing together, and if so, did you test for any effects of different tree species on the growth of the species they're growing with?
So the question is if the species that were growing together in the field, if there were any interactions among between the species. So the species that we particularly planted, they were small enough in size that they are likely not interacting with each other. What it could be interacting with those seedlings is the particular community they were planting after. And in the experiment that I showed you, we weren't looking for particular canopies because we were going to perform these fungicide treatments that didn't work. But when we have been repeating this experiment in the past, we have been selecting different canopies where we are planting the species. But that wasn't part of this analysis. So that's a question I'll be able to answer in the future. Tim.
Later in the kind of complex hierarchical model, was that trying to encompass all of the species, or is that just an example when you're showing the positive or negative interactions? Is that for all of the species combined, or just an example of them?
That was for one particular species. So each species. So was the integrated model of sealant recruitment done for all the species together or each species individually? It was done for each species individually, and each species show a different dynamic. So the one that I was showing you was Caria glabra, but other species have different dynamics.
Yeah, because I kind of was wondering if you would see any kind of trade-off between growth rate and survival, where you have positive, or where you actually have a negative influence of growth rate on survival.
Yeah, so the question is if there were trade-offs between survival and growth, and we did found it. So for some species, higher growth rates were associated with lower survival. And what we think is happening here is because these are first-year sealants that still depend quite a bit on seed reserves, and they may be getting kind of a signal of growing, but if they are growing under an environment that doesn't sustain the metabolism of the whole plant, higher growth rates may be associated with higher mortality. So for a few plants, I can't remember exactly now which species, but I know for a few species that relationship was negative. Okay, back, I don't know your name, so.
I was curious about in the low light conditions, you have a different interaction with the fungi rather than the mycorrhizal, and in the more light, it's positive. Do you have any idea of how it's switching like that?
So the question is how the relationship between mycorrhizal fungi and the plant switches from negative to positive depending on the light environment. So that has been studied for other plants and other systems, and it has to do with carbon assimilation in the plant. So if the plant is able to assimilate enough carbon to, not only for its own metabolism, but also its own growth, and still have a surplus for the fungi, then it's a symbiotic relationship. Both of them gain, they have benefits because the fungi is going to be providing nutrients and water and protection to the plant. The issue is when the plant is not able to fix enough carbon. There is no way the plant can stop this symbiosis. So at that, and under those conditions, the association with the mycorrhizal fungi is more parasitic because it's extracting carbon that the plant needs.
Just a question about the field experiments. I was struck, I didn't realize this, how the difference between the growing season, between slightly up safe and here it's huge, 50%.
50 days, yeah.
Thank you for pointing that out, I didn't know. But with the field work, I mean, the distribution limits of many organisms are set by extreme events. And for instance, this winter and last winter in particular have been more severe than perhaps for the past 20 years. So in your field experiments, how important was seasonality? And you think that the conditions in which they were growing in was reflective of the local biome, that whole area?
Yeah, so the question is, given the differences in length of the growing season between the two sites and the few years of data, if those conditions were representative of the growth. This data doesn't include these last 2 years that were particularly cold, includes previous years. So there are two different things. For example, what is happening in the winter is not really affecting the sealants because the sealants are under, I don't know how many feet of snow. So that's not going to be an issue. Having a very cold winter, it seems to be a really good thing for all these sealants because that means a lot of water late into the season, and recruitment last year was incredible because of the amount of water. So there is a positive effect. The worst year was 2012 when it was extremely dry. So I think the range of conditions that we have seen has been extreme enough to be relatively representative of maybe not the most extreme events, but some extreme events. And we see that in all our experiments, not only these ones. So this is an ongoing experiment. Those sealants are still there. We are continuing studying. This is what happens in those particular years during the establishment process. And another point is, again, this is just establishment of the ceilings, but those differences on growth rates are very important for adults. They may mean the difference between being able to reproduce or not. So we are only studying establishment. So even if the ceilings can establish and can even grow to adulthood, it doesn't mean those are going to be reproductive individuals. So that's one more aspect that is going to be very important too. Ginger.
I'm mostly just curious about this. Did you try to collect all of those seeds? It's sort of difficult to collect seeds in general.
So the question is about where the seed sources were coming. No, we don't. We did not collect the seeds, but we bought it from local seed collectors that collect them in the wild. So we have the coordinates of all the trees they have used and all the areas. So we have a collector that works in the north and another one that works in this area. And we keep track of the sources. So even with the region, we keep track of the different sources of seeds to know if that's making a difference or not.
So there's a lot of genetic variation. There's a lot of variation in the actual genotypes that are going into like the field experiment. So.
Well, okay, is there?
So
I don't know.
I was just thinking about looking at contrasting effects between, say, light on survivorship and growth rates, and are you observing potentially an effective selection, for example? Because if you're measuring growth rates based on who survives, you get to a point where you can actually measure actual growth rates. Could you be seeing selection on that level for different plant genotypes?
So the question is if genetic variability may have been driven some of the results that we see. So I don't know what is the genetic diversity of these species in these populations. So that's the reason why we have different sources of seeds. And we were considering sources of seeds that would be the natural sources of seeds if there is tree migration, the ones in the south and the ones in the north. Another experiment would be to bring them from other different places. There is a lot of variability, but when we look at differences from seed sources, they are not that major. And I don't know what is the genetic diversity of these trees. There is a lot of pollen exchange, so I don't know if it's really playing a role or not. The way I've been going about it, because there's a lot, that would be a lot of work, is to see, well, we are using different sources of seeds. If we see differences in their survival and growth, then we know we need to account for those genetic differences. But until now, we haven't seen anything very strong to make us think we really need to consider where things are coming from. So we consider that as part of our random effects and individual effects that we include in the models. But I still haven't seen a strong sign that will make me think, okay, now we really need to think about the genetics, how different they are, and trying to add this to our analysis. But that's a very good question, and that's another project that we have, is to work with a much larger source of seeds, so we may be able to see those differences, because many of these species grow all the way to Georgia or Louisiana, so why not to bring those sources and see what is the variability in responses of those populations too.
Is there a difference in the explanatory power of different variables that you measured based on how you set up your field experiment?
So the question is if the different explanatory variables have different effects or intensity of effect on the survival and growth of the ceilings. And yes, there are some of them that are more important than others. So for example, growing the habitat versus gap, even if we are measuring light and we are measuring soil moisture, that dichotomy is still very important. And we don't, so we have to include it, even if we don't know why. I mean, it probably has to do with competition for grasses, but we haven't introduced that information yet in our analysis. So there are effects that are very, very important. There are species, for example, that won't respond to soil moisture. And you will think that water is very important for plants. But, some of these species, even in our driest site, they're getting enough water to not make a difference.
Okay, let's thank and congratulate Ines.
Thank you. Thank you.